late can also download the recording. I will post it online. Okay, perfect. So I thought so today I think the main idea is to answer any questions you guys might have about the the lecture on neoplasia. And also, if we have some time, I have some practice questions like case studies that we can do that can help to review the material. So first, please, some students did mention that they have some questions. So please go ahead. You can either type in the chat or ask verbally. Please go ahead. Okay. Nothing pressing. Maybe my lecture was so clear. Okay. Um, then I tell you what, why don't we then start with a tutorial? Just some cases. Okay, we have somebody in the chat that said that they haven't been able to study the material properly because of the exams.
Yeah, so as I understand, they're going through finals right now, so there a lot of people have to wait till the last minute to start studying for our class.
Okay.
So why don't you just do a review right now so we can review the chapter, the students who studied the chapter. Then we can move to clinical case questions to yeah to other questions.
Okay. I didn't prepare a review per se, so I'm wondering how I'm gonna um sorry, I'm trying to share the screen. I'm not sure what it is that you are seeing right now.
We can see your neoplasia
Yep.
lecture.
Okay, is it full screen?
Yeah, well, yeah, it's your browser.
It's in browser. Oh, well, that's a good enough. Okay. Okay, so I can start with a review, but I will just scroll through the PDF that I have. Um so just but it's a long lecture, so I will just go fast through it just to give you the gist of it. Um So, first thing, I went through some definitions. So it's important to know what we're talking about when we talk about neoplasia. So, first of all, neoplasia, what does it mean? It means it's an abnormal growth. But most importantly, what are the characteristics of neoplasia is that it's unregulated, it's irreversible, and it's monoclonal. And that's different from hyperplasia. So hyperplasia is more of a reactive phenomenon that is very well physiologically regulated, and therefore it is reversible. It's also polyclonal. So in the pathoma lecture, there's a very good explanation of how we can assess polyclonality and monoclonality with different tests. Um, and I will not review it. I just made a very graphic picture to kind of grasp. My 12-year-old son understood it, so I figured that's not a bad picture. So in your body, like all the cells have a bit of a different features, and if you have a hyperplasia, each cell will divide into two daughter cells, and then into granddaughter cells, and therefore your hyperplastic phenomenon will be polyclonal, which will it will reflect the variations that you should normally find into normal human cells. If you have a monoclonal proliferation, so your normal cell will undergo a mutation, and that's only that mutated cells that will divide, and therefore your tumor will be composed of all mutated cells that originate from the same mutated mother cell. Very important to use precise terminology because it has different clinical implications. So we cannot just say a patient has a cancer. You have to say what cancer type it is because it will imply different things for the patient in terms of pattern of spread, prognosis, treatment, and so on. Terminology, so you have to know how to call different types of tumors. So depending if it's epithelium, mesenchyme, lymphocytes, melanocytes, for benign tumors of epithelium, we'll add usually the suffix oma. So a benign glandular tumor, we'll call it an adenoma. So adenoma means gland. And if it's a malignant tumor of the epithelium, we'll call it carcinoma. If it originates from the gland, then we'll call it an adenocarcinoma. So again, adenoma means gland, carcinoma means malignant epithelial tumor, and so on. For the mesenchyme, we'll talk about sarcomas. So benign malignant tumor of the fat is a liposarcoma, and so on. Then you can go through all of these terminology. Apologies, I didn't expect to do a review. I was prepared for a Q&A, so I'm not sure how much to go into the detail. So question is, the big question is what causes cancer? So we know that it's genetic mutations that cause cancer. These mutations can be inherited from your parents, or they can occur later in life, and that's sporadic or acquired mutations. And this is the majority of the cases. And sometimes it's an interaction of both. So you have a predisposition to cancer that you inherited from your parents, but then you're exposed to carcinogenic agent that create a double hit. Okay. So we know that carcinogenic agents will cause DNA damage that will lead to cancer in cases of sporadic mutations. And you can divide these carcinogenic agents into broad categories. So I suggest that you go through this list. This is from the Paloma lecture. So it explains different types of well-known carcinogens with their association with cancer. And through my lecture, I give more explanation as well. Uh, Very important is that actually sometimes we're exposed to more than one carcinogens, and these will act as co-carcinogens that really increase our risk of cancer. So a very good example is the concurrent effect of HPV infection and smoking, which really increases the risk of cancers in the lower female genital tract. Okay, so not all carcinogenic mutations will lead to cancer. Sorry, not all genetic damage will lead to cancer. The reason is that some mutations are harmless. They occur in areas of the DNA that doesn't lead to cancer. Sometimes the cell will repair the DNA damage before it can be transmitted. Sometimes the cell will not be able to repair the DNA damage, but will undergo apoptosis, which is a programmed cell death, which is kind of like a cellular suicide. And sometimes the mutations can actually be lethal, so the mutated cell will die and will not be able to transmit the DNA damage through replication. So what we understand from that is that carcinogenic mutations are mutations that are non-lethal, so the cell will survive and multiply and transmit this mutation. They will be harmful, so they will lead to cancer, and they will allow the evasion of protective mechanisms such as DNA repair and apoptosis. Okay, So these are the key regulatory systems and genes that will lead to cancer, um, that will be uh, affected by mutations that will lead to cancer. And through the lecture, I go through all of them. Um, I want you to pay attention to it's a cell schematic of how a cell will divide. So here, that's the cellular membrane in blue. And underneath, it's the cytoplasm of the cell, so inside the cell, and then we have the nuclei right here. Okay, so what happens when a cell wants to divide? So we have first a growth factor that comes. It binds to a growth factor receptor that's at the surface of the cell, and that receptor will be activated, and then afterwards activate some signal transducers that are inside the cell that will then send the signal to the nuclei of the cell, and cell cycle regulators will be in charge of activating DNA transcription to lead to cell division. So any step of the cell division can be affected when we talk about carcinogenic mutations. Okay. Um, okay. So, okay. Apologies. I'm just trying to figure out how much I go in detail. Okay. Um, So this is a normal cell division. So these are proto-oncogenes, and they're in charge of the normal cell division. They're kind of like your accelerator pedal on your car. If one of these become mutated through a carcinogenic mutation, then they become oncogenes, and that leads to self-sufficiency in growth. So it's as if the accelerator pedal in your car gets stuck, and the car just keeps going through the cell division. So I've listed like different examples of oncogenes that can lead to self-sufficiency in growth, and I would like that you go through them. These are very important. I listed the most. There are multiple ones, but these are the most important for your clinical work in the future. Okay, and here's a table that summarized them as well. Okay, please, if you have some questions, like just interrupt me because I didn't prepare to give a lecture per se. Okay. Um, Tumor suppressor genes is an important system that is affected when we talk about carcinogenic mutations. So your tumor suppressor genes, they're like the brake on your car. Okay, so if a tumor suppressor genes gets mutated, it's as if the braking system in your car fails, and your car can, go through the cell cycle divisions without nothing can stop it. And the most important tumor suppressor genes that you need to know about are RB and P53. Okay, so P53 is called the molecular policeman or the guardian of the genome, and it's very extremely important gene to know. So it regulates the cell cycle division when the cell wants to go from the G1 phase to the S phase. So what happens when the cell wants to go from the G1 phase to the S phase is that P53 will be activated, and it will look for DNA damage. And if there is DNA damage, then it will slow down the cell cycle and it will trigger DNA repair. And if the DNA repair fails, then P53 will trigger apoptosis. So basically, if the cell has DNA damage, either that damage needs to be repaired before it can multiply, or the cell needs to trigger apoptosis and die so that the mutation is not transmitted. So what happens if P53 is mutated and not working properly? There is a loss of function, so it cannot trigger cell cycle arrest, it cannot trigger DNA repair, and then the mutations will remain. The cell will be allowed to proliferate and transmit these mutations, and that can lead to cancer. However, you need to have both copies of P53 that are inactivated for tumor progression because if one copy is functioning and available, it can do the job. So p53 inactivation can happen either sporadically, and this is through an acquired mutation, and this is the most frequent phenomenon. So more than 50% of human cancers have actually p53 mutations, and the hallmark of that will be tubo-ovarian cancers, where p53 mutation is really a leading phenomenon. Rarely, some patients have an inherited mutation of p53, so that they inherited it from their parents, and that's called the Li-Fraumeni syndrome. It really increases significantly the risk of having a cancer at a young age. Another way that p53 can be inactivated is by oncogenic viruses. They can bind to p53 and degrade it, and the best example would be HPV, which can do that. Okay, RB, I want you to know about RB. It is very important. So RB stands for retinoblastoma, and just like p53, it also regulates the cell cycle from the G1 to the S phase. And if RB is inactivated, then it's like the molecular brake on your cell cycle is released and the cell can just go through the cell cycle. So same thing as p53. RB can be inactivated by sporadic mutation, inherited mutation, or by oncogenic viruses. This is very important to know. Sporadic mutations in RB, cancers related to sporadic mutations in RB are very rare. They will give rise to retinoblastoma, which is cancer of the retina in the back of the eye. And usually that gives you unilateral disease. It's quite rare. So germline mutations in RB are also rare, but they are the main cause of retinoblastomas. And in this case, it will be familial and bilateral, and the patients will also develop osteosarcomas. So in my lecture, I give you a little bit of a story about Jeff Ely, which is a musician from Toronto, so he's very well known here. So I like to use this story when I teach this lecture to students because, of course, because we're based in Toronto, it really helps students to remember. So Jeff Healy, he was known for playing his guitar on his lap because he was blind, and the reason he was blind is because when he was an infant, when he was 1 year old, he developed bilateral retinoblastomas, and he needed to have both his eyes removed. He was adopted when he was a newborn, so he didn't know his family. So it's at the time of his diagnosis of retinoblastoma that they realized that he had an inherited mutant RB gene that led to the tumors. And later in life, he developed osteosarcomas, and unfortunately, he died relatively young of metastasis from the osteosarcoma. And unfortunately, he did pass the mutation to his son. Okay, important system that you need to know about also that is affected that leads to carcinogenesis is apoptosis. So apoptosis regulating genes, they're a group of genes that will prevent apoptosis in normal cells. So we don't want normal cells to die. We want them to keep, multiplying and building the body. But also these genes will promote apoptosis in abnormal cells. So what we want is if you have a cell that is mutated and abnormal, we want it to go through apoptosis so that it cannot transmit its DNA damage. So we just saw that p53, not only is it a proto-oncogene, it's also an important pro-apoptotic gene, right? Another important pro-apoptotic gene is BCL2. So you need to know about BCL2. It's a very important one. You will hear about it a lot in your practice because BCL2 is overexpressed in a majority of follicular type B cell lymphomas. Okay, DNA repair genes. So like we're constantly exposed to carcinogenic agents. There are defects in transcripts also that happen when the cell divides. So we actually have like quite a few mutations in our body. Um, Most of them are not dangerous, actually. Some can be, but we have this mechanism of repairing the DNA when the cell divides. The problem is if this mechanism is dysfunctional, then mutations cannot be repaired and then they can accumulate. And if they accumulate in oncogenes or tumor suppressor genes, then that's where that can lead to cancer. So very important DNA repair genes that you need to know about are MMR DNA repair genes. There are a group of genes. The most important are MLH1, MSH2, MSH6, and PMS2. And they really play an important role in the production of colon cancer. So there, we have acquired mutations in these genes in up to 15% of sporadic colon cancer. Um, and also we can find inherited mutations in these genes, and that leads to what we call Lynch syndrome. So Lynch syndrome, it's really the hallmark of inherited predisposition to cancer through a defect in DNA repair. It's a very important syndrome to know about. It increases the risk significantly of colorectal cancer, but also other types of cancer. So endometrial, ovarian cancer, gastric cancer, breast cancer, and so on. And not only the patients have an increased risk of having cancer, but they develop it at a very earlier age compared to the general population, usually in their 40s.
Hey, Doc, if I could add something real quick, could you scroll up one slide? Um, yeah, so this section, these two slides, these are not in Pathoma, so just write this down that this is something you need to add to your memory because it's not in there. So, but obviously she's putting it in her lecture, so it's important. Obviously, having defects in DNA repair is a big deal. So please make sure that you memorize these two slides, including Lynch syndrome.
And you will go through this also. I would expect in your next semester when you go over GI cancers, but it is very important material to know. Okay, so how do we diagnose Lynch cancer? So the diagnosis is through genetic testing. So you need to prove that the patient is the carrier of a known mutation that leads to Lynch syndrome. But if the testing is not available, there's other tools. So personal and family history can be very powerful. So if you have a family where there's multiple individuals that are diagnosed with a cancer that has been associated with Lynch, so you have like many people with colon cancers, some with endometrial cancers, and diagnosed at an earlier age than the general population, then that's a big flag that maybe there is Lynch cancer, Lynch syndrome in this family. Or if you have one patient that has had more than one of these cancers, also it's a big flag. Another tool is when a patient is diagnosed with cancer and the pathologist receives the tissue, then if it's a cancer that is known to be associated with Lynch syndrome, we will test it using, we will test the MMR genes using immunohistochemistry. And if there is an abnormal expression of these genes on the tissue, then we will flag the patient that, there's a possibility of underlying Lynch syndrome and that patient needs to be seen by genetics. Okay, so if the patient has Lynch syndrome, what does it mean? It means that they need to be screened. Okay, so they need to be screened at an earlier age than the general population. So, for example, to start annual colonoscopy in their 20s or 30s, and so on. And some patients can even undergo preventive surgeries. So, for example, they can have a colectomy to prevent the occurrence of cancer. The ladies that have Lynch syndromes, we would recommend that they undergo a hysterectomy after they have completed their family to make sure that they don't develop endometrial cancer. Okay, telomerase reactivation. So we know that the cells can divide. They have a limited capacity to divide, so 60 to 70 times in their lifetime. And once they reach this capacity, they become senescent. And how does it work? That's actually relatively new knowledge. So it was discovered that the telomeres, there are a short section at the end of each chromosome. And every time that a cell divides, that section shortens. The telomeres shorten. So once it becomes too short, the cell cannot divide anymore. So what happens that cancer cell can reactivate the enzyme telomerase, which will regenerate the telomere, so lengthen them back. And that will allow the cancer cell to keep replicating. Okay, angiogenesis is an important system also. So tumor cells are like any cells in your body. They need oxygen, they need food to grow. But once the tumor keeps growing, eventually there's not enough blood flow to feed it. So for the tumor to be self-sufficient, what happens that cancer cells acquire the ability to secrete angiogenic factors. So these factors, for example, VEGF, will stimulate the growth of new vessels that will feed the tumor. It's a double whammy because also the problem with that is that the tumor will increase its access to the blood supply, and that will increase the risk of metastasizing. Since we know about this phenomenon of angiogenesis, it has led to the production of drugs that will inhibit angiogenesis. An example is bevacizumab.
Hey, Doc, if I could add one thing. So, whenever you guys think of patients with cancer, you always think, that they get tired, they're sleepy, they have low energy. And sometimes it's because of the drugs we give them, but also it's because of that concept of angiogenesis and the extreme amount of, essentially, I always tell people, like, when you're having a baby, that's almost like a controlled, you know, situation where the body is doing all these same things, where it's making blood supply, where it's doing this. And if you notice, pregnant women, especially later on in pregnancy, they're a lot more fatigued, things like that. And this is the same reason. The tumor takes up a lot of energy. A lot of times, if it gets large enough, it diverts a lot of blood flow. Not clinically significant where they're dropping their blood pressure, but to the point where, they require higher nutrient consumption. So just something to be aware of if you have that kind of a clinical scenario that's sometimes given, especially on tests.
Okay. Important system is immune surveillance. Okay, so tumor cells can develop the ability to evade immune surveillance because technically a tumor cell is abnormal. So it expresses abnormal protein on its surface, and then the T cells can recognize it and see it as a foreign object and actually target it for destruction. An efficient tumor, an efficient cancer, should have the ability to evade that immune surveillance. And an important mechanism you need to know about is the down regulation of MHC1. Okay, so MHC1s are molecules that are crucial to present antigens to the T cells so that the T cells can uh, uh, target cells and destroy them. So some cancer cells have developed the ability to downregulate MHC1s, and that camouflages them, that makes them less visible to the cytotoxic T cells, and then they can evade the immune system better. There's other mechanism, but the downregulation of MHC1 is the most important one I want you to know about. And finally, important thing is that cancer cells need to develop mechanism that will give them the ability to invade and to metastasize. Okay, so here we have like a schematic. You have like epithelial cells. They sit on a basement membrane and they're bound to each other using adhesion molecules that are called cadherins. Okay, so in order to invade, cancer cells need to downregulate these cadherins, so detach from one another, and then they need to attach to the basement membrane. So they will develop receptors to attach to the laminin that's present in the basement membrane. And then they will develop, they will secrete collagenase, and collagenase will digest the collagen in the basement membrane that will allow them to breach the basement membrane. And afterwards, they will attach to the fibronectin in the underlying tissue and will allow them to pull themselves into the underlying tissue and invade. And once they have breached that basement membrane and are located in the underlying stromal tissue, they can have access to lymphatics and blood vessels, and that will help them to spread throughout the body. Okay, any questions so far? Not sure I see the chat. Okay, let's just continue. Okay, this is how a tumor, so more the clinical characteristic of cancers, how does it grow, how does it spread? So we went through the more molecular phase of it, right? Now let's just go through the more clinical aspect of cancer. So how does it grow? How does it spread? What does it look like? How do we deal with it? And so on. So for a tumor to occur, first you need to have a transformation. So we said the normal cell needs to transform into a neoplastic cells, and then it needs to grow, and eventually it will form a tumor that is visible to the naked eye, and then it needs to acquire the ability to invade and metastasize. So benign tumors will go through that transformation and growth phase, but they do not have the ability to invade and metastasize. While malignant tumors or cancers can develop this ability to invade and metastasize. And that's the very, very, very important point. What's the difference between a benign tumor and a malignant tumor? Okay, is that malignant tumors have the ability, can have the ability to invade and metastasize. So they have this potential, while benign tumors theoretically do not. Of course, there's exceptions to that, and I will not go through it, not to confuse you. Okay, so these are the two important points. So in ability to invade and metastasize, but there's also different clues that you can use in clinic to try to figure out, okay, is this tumor more likely benign or more likely malignant? So benign tumors will usually be slower growing and better circumscribed, so and more mobile, while malignant tumors, cancers, can grow faster, can be more infiltrative and more fixed to the surrounding tissue. And I give this example of a uterus. So the wall of the uterus is made of smooth muscle. And if you have a benign smooth muscle tumor, it's called a leiomyoma. And it's usually very well circumscribed. So the edge of the tumor is very well defined compared to the surrounding uterine wall. And actually, there's a cleft usually in between. And the surgeon can use that cleft to actually put their fingers or tools in the cleft and scoop out the leiomyoma. While with the leiomyosarcoma, the edges are ill-defined. Okay, so it's infiltrative into the myometrium. Um, and you really cannot like just resect it. So if you want to remove a leiomyosarcoma, you have to remove the entire uterus. So do a hysterectomy. Okay, so having said that, these are clues, but at the end of the day, who makes a diagnosis of tumor and cancer? You need a tissue sample to, and it's a pathologist that will make that diagnosis. So either through a biopsy or through an excision. And how does a pathologist make that diagnosis? Well, we'll look at the sample, at the cells, and there's different types of different features that we'll look for to make the diagnosis. So first, compared to normal tissue, a tumor will be more disorganized, abnormal nuclear shape, hyperchromasia, which is when the chromatin becomes very dark, and we can find mitotic activities also. All of these features, you can find it in benign or malignant tumors, but they're much more pronounced in malignant tumors. So I just put a picture to kind of give you an example. Okay, this is the normal uterine wall. So the cells are elongated. And this is a benign leiomyoma. And it looks relatively similar. It's just a bit more dense, a bit disorganized, and that's it. And this is an example of a leiomyosarcoma. So you can see that the cells are much more abnormal looking, big nuclei, small nuclei, very dark looking cells, abnormal mitotic figures here. Okay. Having said that, when I speak to the clinicians, I cannot tell him, well, um, sorry, let me go back a little. Okay, so here it is. Okay, that leads us to talk about differentiation. So what is differentiation? It's how much does the tumor cell, look like the cell of origin? Okay, so some tumors can be well, moderately, or poorly differentiated, depending on how much they look like the cell of origin, or they can be anaplastic, which means they have no resemblance with their cell of origin. Benign tumors, very important, they're always well differentiated, while malignant tumors can be well, moderately, and poorly differentiated. And that's very important to know about because the level of differentiation usually has a prognostic significance. So a better differentiated tumor has a better prognosis than a poorly differentiated tumor. And I've just put pictures here to give you an understanding of this is a normal colon made of glands, okay? And this is a well-differentiated adenocarcinoma. So adenocarcinoma, malignant tumor of the glands. So you can see that you can still identify some gland formation. They do look like the original glands of the colon, so this is a well-differentiated adenocarcinoma. It is a bit disorganized because here, for example, you have three glands that merged into each other. So it is abnormal, but still looks like the normal colon. In comparison, that's a poorly differentiated adenocarcinoma. So only focally I can see a bit of gland formation. And that's an anaplastic carcinoma. So I cannot tell that originally this was a gland. I will need special studies to help me out with that. Okay, so having said that, I cannot just tell the clinician, well, it looks bad. It looks not too bad. Uh, so we need an objective system for pathologists to convey to the clinicians how bad does the tumor look. And that is what we call grading. So grading is an objective system. And it's a very crucial component of the pathology report. It will guide treatment. It will give information about prognosis. There are different grading systems that we use for different organs. Here are a few that you need to know about. So the Gleason score, for example, is a grading system we use in the prostate. So basically, the pathologist will look at the tumor, the prostate cancer, and will quantify how much of the tumor is glandular. And if it's mostly glandular, then you have a low score. And if it's mostly solid or different types of formations, then you get a higher score. And then it's a bit of a complicated system where you have to add up the different patterns and you get a final score. Another example will be breast cancer. So breast cancer will use a Nottingham score where we will assess the architecture, the pleomorphism, which is how bad do the nuclei look, and the amount of mitosis. And each one will give a score and we'll add this up for a final score. There's also different WHO grading that we'll use for different organs. Okay. Perfect. Okay, so this is not as important. We'll go through it.
Yeah, Doc. I mean, I think for the sake of time, if there's anything important, go through it. Otherwise, we can just open it up
Yeah,
to questions. If the students
yeah,
have any questions, they can start sending it now.
yeah. So, but I think maybe, yeah, for the sake of time, we'll just stop here. So please go through the lecture. It's more detailed, um, as well as the pathoma lectures. So let me just.
Yeah.
Okay.
All right, let's start with any of the students. Do you guys, anybody here have any questions? I want to start with that first. All right, let's see. Muhammad raising a hand. You can go ahead, Muhammad.
Go ahead.
No.
I think you're on mute if you're trying to talk, please.
Oh, do we have to
Oh,
unmute?
I need to unmute. Oh,
Oh, I don't know how you do that. No, because
sorry, I'm not. This is somebody, this is my colleague's Zoom meeting space. Like I'm not used to do that.
Muhammad, you can't unmute yourself because I'm a participant too, and I just unmuted myself. And I think other students have unmuted themselves too. Okay. So
You
he's
can
gonna write a
write also the
question.
question. Okay. So there was a question about in the chat about retinoblastoma, asking can it occur in both eyes? Yes, definitely. Um, so usually if it's a sporadic mutation, it will be exceedingly rare to have it in both eyes because you need both retinoblastoma genes to be mutated to develop a retinoblastoma. Okay, so for that event to occur in both eyes, it's not a frequent event and like that would be exceedingly rare. But if somebody has an inherited mutant RB gene, then already all of the cells in the back of his retina have one non-functioning RB gene and one functioning. So you just need another hit to the functioning RB gene to develop a retinoblastoma. So yes, if it's an inherited phenomenon, then the patients do develop familial bilateral retinoblastoma.
Yeah, and I think high yield guys, because I'm looking at what questions we're making is the Knudsen two-hit hypothesis. Um, so, and I put up a photo in the chat of the loss of red light reflex. So sometimes that'll come up in exams where, you take a light, the light that they use and they look at your eyes on children. A lot of times if you're doing your pediatric rotation, you're going to be doing the screening and you're going to see, look, one light, one eye, you have the loss of red light reflex. Like it should look, when you take a camera, when you take a photo, sometimes that's how people discover it too. A lot of times people have like a family photo and they'll say, hey, look, everybody's eyes turn red in this photo, but that one kid, his eyes, only one of them turned red. And that's sometimes how we discover it as well. So high yield for a lot of board exams.
Another question in the chat about what other mechanisms contribute to increased mobility and invasiveness of tumor cells? So yes, downregulation of E-cadherin, like you said. So basically the cells will downregulate and lose some of their junctions. So between one another, so they can invade better. Then other mechanism, they will develop receptors to grab on to the basement membrane, which is laminin receptors. Third mechanism is to develop the ability to secrete collagenase, which will digest the collagen in the basement membrane. And fourth mechanism is they will develop receptors to fibronectin. So fibronectin is located in the underlying stromal tissue, so they can grab on. Once they have digested the basement membrane, they can grab on to the fibronectin in the underlying tissue and pull themselves into the underlying tissue.
Yeah. I think also a good example that also comes up, and I don't know if any of you have this, is lipoma, right? So I have one, let's say right here. Actually, I have one right here. So it's round, it's well circumcised, right? It's freely mobile, so there's no binding down, right? So if something is malignant, it's going to be, I mean, this is not a perfect example, but it's going to be a little bit more, tied down to the bottom. And so in my mind, I always remember, okay, if it's doing collagenase activity, if it's getting, a significant amount of angiogenesis, it's going to be, stuck down. I don't know if that's actually true, but this is the easy way to remember.
Because if it was a liposarcoma, it wouldn't be very mobile because it will,
Yeah.
have these finger-like projections to spread, to attach to things. and you couldn't move it like that, right?
Yeah.
It would
So
be
here,
attached to
moving it
pull
around.
itself.
This is benign, benign.
But you need a biopsy to prove
That's true. I mean,
it.
I have a lot of them. I would take a lot of biopsies. Um, okay.
How
Anybody
the
else?
hit occurs. Okay. So I guess you mean
Double hit. He wants to know.
the double hit, yeah. So that is like sporadic mutations. Okay. So they will occur through you're exposed to a carcinogenic agent that will lead to a damage in your DNA. Okay, so that's one thing. It could be just when the cell replicates, it has transcription errors. It makes an error in the transcription, things like that.
Okay. And if none asks, is it easy to examine the retina in children? Or do we need, well, for the sake of your exam, it's easy to diagnose, okay? You just look at the red light reflex. But sometimes in real life, um, you do a dilated eye exam. You don't just assume, because there's other things that can cause the loss of red light reflex.
I'm not a clinician, but I know that some parents actually bring their child because the child is looking at the TV and the parent is on the side and notice that there's a different color, different
Yeah.
reflection in the eye of their kid. And they're like wondering what this is, and that's how, they're diagnosed with retinoblastoma. So I would expect it's not that hard to examine.
Yeah, and the example I gave of the, you know, photograph, that I've seen that before, where somebody
Mm.
pick it up a lot of times on photographs. Okay.
There's a question in the chat that I'm reading.
Okay. Thank you.
Wait, I'm trying to read it.
Regarding the interplay between
Okay, I didn't go into into detail into that. Uh, and you catch me at a blank. I am a gynae pathologist. I have forgotten about that.
well, I mean, I think just to make it simple, right? So um, he's asking essentially, if I'm getting it right, if a cancer cell has CDK4 mutation leading to amplification in uncontrolled cell cycle, how would this specific oncogene activation potentially circumvent the normal suppressor function of RB and P53? Um,
So I know that RB needs to have the ability to phosphorylate and phosphorylate itself in order to work and stop and go. And that's the cyclin D1s and CDK4 complex that will help doing that. So if that mechanism is not working, then RB will be stuck into the state that it keeps going, or it cannot stop, sorry, the cell cycle. And that would lead to uncontrolled cell growth. That is my understanding of it. How deeply do you have to go into it? Um, I think it's basically to know the general principles. I wouldn't go that deep
I
into
think
it.
that's as deep as it should go.
Yeah, like this is not a research or a PhD exam, right? It's more to know the general principles.
I mean, even when I was in med school, a lot of this stuff is new, because this wasn't discovered until not too long ago. So sometimes this is all the depth we have.
Like as pathologists, we use P53 and RB every single day. We test for it and everything. But do we go in depth into the mechanism of it? No.
Okay. All right, guys. Keep the questions coming, and feel free to talk. We like hearing your voice. It's okay.
So again, this is not my Zoom meeting, so I don't know what happens at the end of the time. I don't know if it stops or like this is my colleague's Zoom space. So I
Oh,
hope we won't be disconnected. But
we
it could
can,
happen.
we can send another quick link if it does, and people still have questions. Um,
I just don't know if my colleague needs his Zoom space.
yeah.
That's
Um,
not true. But if you have questions, please feel free. I sent you my email. You have the chats. Um,
there's a few. As I was going through the questions, there's a few high yield stuff, guys, that I think is important. And again, I'm not a pathologist, but I know that this comes up over and over and over again. So chapters 1 through 3, they're gonna come up in every single section. And I think that's why even when I was studying pathology, these were the hardest chapters, chapter 1 through 4, these concepts because they come up everything. So I know that a lot of the questions are going to be based on table 3.2. That's going to come up a lot, and I know Dakshi put it in there as well. 3.2 is all the carcinogens associated cancers. That's just memorization, unfortunately. Some of it's conceptual, but that comes up in a lot of the questions that we're going over. Um, Trying to think if there's any other main things. Obviously, she covered all the oncogenes, the tumor suppressor genes, the regulators of apoptosis, but table 3.3, again, it's something that you learn but sometimes forget. But if you guys go in your book, table 3.3 again is there's a lot of stuff in there, especially if Dr. Sattar highlights it in his lectures as well. It comes up quite a bit, like the HER2/neu, um, and the all the oncogenes and the associated tumors. Um, three
there's a
point.
few chats in Arabic. Sorry.
Oh yeah, yeah, go ahead.
If you can now, if the students can, somebody can translate them in English. عشان أنا
Yeah,
مش بتكلم عربي كويس يعني.
I was just gonna I'm just gonna use my Google Translator.
Don't read it. Okay.
I don't know.
والعربي من فلسطين صعبة شوية. أنا عارفة العربي من مصر ولبنان بس فلسطين صعبة شوية. That's the extent of my Arabic.
I think
All right. So I think we'll, unless somebody has a pressing question, I think we'll stop here. Thank you so much for joining us, and please do not hesitate. Send questions by email, by chat, whichever works for you.
Okay, and everybody, the Google Meet for the next lecture for the next Dr. Bassam is starting now as well. Okay, so you should check your WhatsApp. The Google Meet link should be in there. All right, so I'll leave
All
this on
right.
there.
Thank you.